Good afternoon, everyone. Today marks the first day of our e-facilitation, and we'll be looking at the introductory lecture. In BIO191, we're going to introduce you to the course, what is expected of you, and create awareness that the practical exercise will start next Monday for you to go to your respective centers to attend. So at this point in time, if I may ask, what is biology from your secondary school knowledge?
Biology is the study of life. I think that is
Good. Very good. Biology is the study of life, and by life, we mean both plants and animals. You may begin to wonder. For art subjects, history, literature, English, probably when you finish the lecture in school, in class, you go and speed up for your exams. But for sciences, it's not like that. After teaching you in class, you still go to the laboratory to perform that which you've learned in class. So at this juncture, I will start sharing my slide for us to introduce the course. Just as I said earlier, that what we have today is introductory lecture. Meanwhile, I'm Professor Maureen Chukwu. We are facilitator for BIO191, and I'm from now headquarters at Abuja, Faculty of Sciences. I'm the Deputy Dean. So let's proceed. Introductory lecture. On completion of this lesson, you'll be able to state why practicals are important in biology, examine how to successfully carry out practical work in biology, state the involvement in practical biology, and make good biological diagrams. Let me minimize this for us to have full view. The importance of biology, practical biology, just as I was explaining, that as students, after being taught in class, go and read and subsequently sit for their exams. But for sciences, it's not like that. After the lecture, you still go to the lab, see the things you've been told of, practicalize, see equipment, see specimen, as the case may be, handled them. And all this is to ensure better understanding of the subject. It enables discovery of facts about organisms, enables scientists to approach problems in a logical, orderly manner, and provide solution to the challenges of man. Quickly attempt this activity. What are the involvements in practical biology? Let's now see them from our slide and further explanation. Practical biology involves the following: field study, there are times when we have to go to the field, natural habitats of organisms, to watch them, how they live, how they interact, and so on and so forth. Preservation and storage. Preservation and storage, I agree with you. Parts of the organisms to watch them, how they live, how they interact with other organisms, the factors, both biotic and abiotic factors. And there are times we have to collect specimen. When you get there, you may need to collect specimen that's live organisms, as the case may be. Then have to also revert to the lab. In the lab, those specimens may need to be preserved and may need to be stored, as the case may be. Then lab technologies takes care of those ones and will even introduce you to them. Then there are times you may need to prepare specimen for lab studies. Yes, temporary slides, you prepare them for lab studies, use and wash off at the same time. Then there are times when you need to make sessions and slides. By sessions, you need to cut open. You cut open when you want to look at the internal structures of the organisms. Cutting open can be longitudinal from head to down or transverse across board. We're going to look at all of them. When I talk about biology drawings, in this course and the practical session, you will encounter various drawings. And there is a way that biological diagram should be drawn for it to be okay. First, you can see two diagrams displayed. One is with straight line, and the other one will curve and curvy and woolly. It should not be. It's the straight one that is marked good that is the correct one. And to be able to achieve this, you need to come into the lab with sharpened pencils, not just one, more than one, so that if one breaks, you have a substitute immediately. Your power of observation has to be very sharp. Make clear-cut observation, follow instructions as given. Even when it comes to experimentations, instructions need to be followed systematically so that you won't make mistake and get the correct results. Then you draw with continuous thin line, not really as seen in the other diagrams. As I was talking about, views, we have to got the view. View simply means from which side of the specimen are you drawing. If you are drawing from the front, it is the anterior view. Drawing from the back, it is the posterior view. Drawing from thumb, it is the dorsal view. Drawing from down beneath it is the ventral view, and drawing from the side is the lateral view. See figures. And figures have to be well put up. Figure 1.2, a dot. The other one seen earlier, I can see is figure 1.1, a leaf drawn with fuzzy or woolly lines, and a clean, clear drawing of the leaf. Then we now have, having explained the views, side we are drawing from, let's look at sections. As I mentioned earlier, that section is being cut open to see the internal structures of the organisms. It can be longitudinal, cutting from top to bottom. Like here display longitudinal section of a mango fruit, see the inside. It can also be transverse. Okay, see, I won't fail to talk about magnification here. Longitudinal section of a mango fruit times 1, meaning magnification. And magnification simply means how big is the fruit against how big is your drawing compared with the specimen itself. Are they of the same size times 1? More than bigger than the drawing, twice, half, times 2, times half, as the case may be. Then, drawing, if you cut transverse is cutting across. What you see is the transverse section. By this date here is transverse section of a garlic roots. You have to put magnification there. This is the internal structure of a garlic root cut transversely. Then labeling. Having put up your diagram, how do you label? You label with straight line, draw with ruler. We call them guard lines. Label one side if we don't have many structures to label. Both sides if there are many structures, but note that the guard lines must not cross each other. They must not overlap. And they should not have arrows at the edge. If there are arrows, the meaning changes. It becomes direction, no longer guidelines. See labeling here. Method of labeling. That rabbit there, well labeled. But the amoeba, you can see the drawings overlapping, not well labeled. Then we talk of headings. That's how do we name the heading of our figures. First of all, you identify the figure. As we saw a mango. Then we talk of the view, side which you are drawing from, and finally magnification. As we've already seen, I think lateral view of a dog, anterior view of a dog, times 1 or 2 depending. Identity means the name, the view, the side you are drawing from, and magnification, how many times. And finally, talk of experimentations. As I've already told us, that there were times when we've got to be in the lab and perform experiments. And experiments can be to prove facts that are already existing or to verify them. And by the time you conduct your experiments, you've got to report specific way of reporting of our practical work. First of all, we are supposed to put the dates of which the experiment was carried out, put up the purpose of the experiment, materials used for the experiment, procedure, results, and conclusion. For example, maybe an experiment to show that oxygen is given out during photosynthesis. Having written your dates, you know today's date, then materials, maybe water plants, elodea leaf, conical flask, funnel, and so on and so forth. Then procedure. You put your water into a beaker, then you put a water plant there, say elodea plant, then cover a funnel with water, distilled water, and invert it over the plant, turning it upside down, and then cover with a test tube. And find out that as the time, once it's under light, bubbles will be coming up. And that bubbles, when tested with glowing states, will light up, showing that it's oxygen. And I want to introduce us to an instrument called microscope. Microscope is what we'll be using more often in this course, because I'm going to use it to view microscopic organisms that normally naked eyes will not see. So this is what is involved. Then we have the ocular, that's the eyepiece through which we view. We have the nose pieces of various magnification. We have 10x magnification, 40x, and 100x. As magnification increases, the size of the organisms or whatever that is displayed also increases. We have the arm for carrying the microscope, and we have the base. Carry the microscope with the base and the arm supporting. Quartz focus and the fine focus for focusing the objects you are viewing, be it in your slide. First, you start focusing with squash focus, that is squash adjustments. Then when you must have seen it, you use time adjustment to make up. And we have the power switch, which in this case is the light we'll be using. We have the stage where we put our slides. Then we have the projection lens. When caring for the microscope, we always clean our microscope, the lenses, with lens tissue. Carry with 2 hands for care. Make sure that everything is in order. I have to take care of my microscope more so because it is a very expensive instrument that caution must be taken not to damage. In conclusion, in this session, we've learned that practical work is an essential component of theoretical study of biology. The basic essentials and techniques, practical biology, you've learned it, the things you need for practical work, and the need to exert careful and follow instruction during practical work. Then, answer this question at your own spare time. Draw and label a compound microscope. Outline the functions of any 6 labeled parts. Thank you. In our next class, that's next Monday, please same time, we'll be looking at procuring plant and animal materials for laboratory work. Thank you so much for taking your time, giving your time for listening and your patience. Now I'm going to stop sharing. We will converge so that we ask our questions. Yeah. Any question? Any question? You can ask your question. This is time for questioning.
Thank you very much, ma'am. I have a question concerning the guidelines, that is labeling. You share the amoeba and the because I just need you to throw more light on the labeling part so that I can understand more, because looking at it just looks the same to me, so that I was able to understand the correct way to do them.
I was trying to show you how we label, that the labeling lines should not meet each other, should not overlap or cross each other. In that of amoeba, okay, let me bring it up again. You can see the labeling lines crossing. That is why it was marked wrong. But in the other rabbits, there was no crossing of the labeling lines. Let me open it again. Yes. See amoeba. This nucleus and cytoplasm, the lines are crossing each other. That is what made it to be wrong. But here, everyone is standing alone, parallel to each other. They should not overlap. Right? Are you there?
Yes,
Any other question? Any other question before we call it a day?
thank you very much, ma'am.
Any other question? No. So please, let's make it a date to let us know, to let our classmates know that this class, online class, will be holding every Monday, 12 to 1 p.m. And as we do that and come, we'll be having more knowledge of the subject matter. And also, I'd like to make it clear that the practical, this is a practical course. I think that from this year, it is even the performance of your practical that will be used as your assessment score. So by Monday, practical will start. Though starting with 192, not 191, a designated center at every geopolitical zone. Make it a date to attend in your own time, depending on the set you pick. 2 weeks per set. Thank you very much. Meanwhile, I've opened discussion forum for the week, and you are expected to go and put your input. Thank you very much for your attention. So,
Thank you.